Topic 5 Energy for Biological Processes 5.1 Cellular Respiration. Core Practical 9 Investigate factors affecting rate of aerobic respiration. This could be done using an equipment called a respirometer, and what the respirometer measures is the uptake of oxygen by organisms that respire. When these organisms respire, they produce carbon dioxide. And that carbon dioxide could affect the results of the experiment, so you need to be absorbed, and this is done using a chemical called soda lime. And therefore, any changes in the volume of the respirometer is due to the uptake of oxygen. The carbon dioxide that is produced from respiration has no effect. As organisms use oxygen in respiration, the pressure decreases, and this can be seen in the manometer, which has a colored fluid, and that colored fluid then moves towards the tube containing the organisms that respire. The amount of movement or the rate of movement of the liquid can then be measured in the scale, and that measures how much colored fluid moves can be used to calculate the intake of oxygen per minute. So the example of this unit would be centimeter cubed per minute. A syringe can therefore also be used to return the colored liquid in the manometer back to its original position, and therefore you could do repeats. You could also make changes to experiment, so you could see the effects of different independent variables, such as the effect of temperature on respiration, or the type, mass, or number of organisms, how they affect respiration. So using the syringe means that you could do those repeats or change the independent variable. If temperature is not an independent variable, that means it has to be controlled, and one way to keep the temperature constant is by using a water bath. In this experiment, you have to take into account the safe and ethical use of organisms. That means the organisms have to be protected from the soda lime, and this is because the soda lime is corrosive, and this requires the use of a barrier. The experiment that is being done causes the colored fluid in the manometer to move towards the tube containing the organism due to the organism respiring. And to check that it's the organism that's causing the movement of the colored fluid, you can do the experiment as a control experiment where no organisms are used, and therefore this should result in the colored liquid movement not occurring. And that's proof that the movement of the colored liquid in the manometer must be due to the organisms respiring. So this is a control experiment, not a control variable. In topic 2, we learnt how to draw and label a mitochondria, and you can see that there is an outer and inner membrane, and the inner membrane can be folded inwards to form the crista. There are also ribosomes in the mitochondria and the matrix, and it also contains DNA. State different stages in aerobic respiration. The first stage of aerobic respiration is glycolysis, and this occurs in the cytoplasm. After glycolysis, there's the link reaction and also the Krebs cycle, and both occur in the matrix of the mitochondria. And then to finish off, there is oxidative phosphorylation in the inner mitochondrial membrane. Describe glycolysis in cytoplasm. So the reactants to start off with is glucose, which is the monosaccharide, and it is then phosphorylated using ATP. The product is then converted into 2 glycerol 3-phosphates, and those 2 glycerol 3-phosphate molecules is then converted to 2 pyruvates, and this also produces reduced NAD, which is known as NADH, and ATP. From chemistry, you should have learned oil rig, where oxidation is when there is a loss of electrons, but this could be also the loss of hydrogen, whereas reduction is gaining electrons or gaining hydrogen. And this is the opposite for oxygen. So oxidation is when a molecule is being oxidized by gaining oxygen, but in reduction, it's a loss of oxygen. In glycolysis, we could see that NAD has gained hydrogen, therefore it's become reduced. Because reactions are coupled, if one molecule becomes oxidized, the other molecule must become reduced. So if NAD has gained hydrogen and become reduced, therefore the molecule that gave the hydrogen must have lost it and therefore become oxidized. So we could see that glycerol 3-phosphate has lost the hydrogen and therefore it's been oxidized because it's a coupled reaction. Describe anaerobic respiration. When cells don't get enough oxygen, the products of glycolysis, which is pyruvate, can't go to the Krebs cycle, which is the aerobic stage of cellular respiration that occurs in the matrix of the mitochondria. And this is because there is no oxygen available as the final electron acceptor, which you'll understand in further slides afterwards. Therefore, there's no NAD available as all of the NAD eventually becomes reduced. Anaerobic respiration takes place and the pyruvate is reduced to lactate by oxidizing the NADH, which is reduced NAD. Glucose is partially broken down and this produces limited yield of ATP in the absence of oxygen for anaerobic respiration. The toxic lactate is oxidized back to pyruvate, which can then enter the Krebs cycle for aerobic respiration when there is oxygen available. And the amount of oxygen that is required to break down the lactate so that it could be oxidized back to pyruvate is known as oxygen depth. This occurs when oxygen is then available. Explain how lactate as byproduct of anaerobic respiration affects muscle contraction. The lactic acid dissociates to form lactate and hydrogen ions, which increases during anaerobic respiration in muscles. This causes the pH to decrease and muscle tissue becomes acidic, which reduces the ability of muscle contraction, which is known as muscle fatigue. An alternative idea is that the movement of lactate and hydrogen ions into the blood lowers the pH of the blood, and this reduces the nervous stimulation from the central nervous system, and then that stops muscle contraction. Anaerobic respiration in plants and fungi is different to mammals, and here the glucose is partially broken down into ethanol and carbon dioxide with limited amount of ATP being produced, just like anaerobic respiration in mammals. When there is enough oxygen available, the pyruvate produced from glycolysis is transported to the mitochondrial matrix. And the mitochondrial matrix is where the link reaction and Krebs cycle occurs. So we want to explain the complete oxidation of pyruvate. This is when there is oxygen available, it's aerobic respiration. And here, the link reaction oxidizes pyruvate to form NADH, which is reduced NAD. Carbon dioxide is also removed, and the molecule combines with coenzyme A to form acetyl-CoA. That acetyl-CoA then goes to the Krebs cycle, and in the Krebs cycle, acetyl-CoA, which is a 2-carbon molecule, combines with a 4-carbon sugar to form a 6-carbon sugar. And that 6-carbon sugar then gets decarboxylated, which means that there's a removal of carbon to make carbon dioxide. It then gets oxidized by NAD and FAD, which are electron carriers. So NAD and FAD gets reduced. ATP is also produced directly in the Krebs cycle. So explain the role of Krebs cycle. It completely oxidizes pyruvate or acetyl-CoA of glucose, and that releases as much energy as possible to generate ATP directly. You also produce NADH, which is reduced NAD, and FADH2, which is reduced FAD. And those electron carriers can then be used to produce ATP in the electron transport chain stroke oxidative phosphorylation stage of aerobic respiration. Do note that in glycolysis, the glucose is broken down into two pyruvate molecules. So therefore, in the link reaction, those two pyruvate molecules will convert to two acetyl-CoA molecules. So if you look at the Krebs cycle, this is for per acetyl-CoA or per pyruvate. And if you want to know per glucose, you'd have to double the number of carbon dioxide, reduce NAD, reduce FAD, and ATP produced. Those numbers would be doubled for per glucose. So for one Krebs cycle, there's two carbon dioxide molecules produced, but then for per glucose, there'd be 4. For per acetyl-CoA in the Krebs cycle, there's 3 NADH, reduced NAD, and 1 FADH2, which is reduced FAD, but those numbers would then double per glucose, and the same would happen with the number of ATP, that would also double per glucose. So here's an animation that shows what happens to the reduced NAD and reduced FAD that are produced. And because they're reduced, they can then be oxidized, which releases hydrogen ions and electrons. And these hydrogen ions can then be pumped across the membrane against the concentration gradient, whereas the electrons are transported from one electron carrier protein to another on the membrane. And as they get transported, they provide energy for hydrogen ions to be pumped across the membrane against the concentration gradient. The electron is then finally accepted by oxygen, which reacts with hydrogen ions to produce water, and the hydrogen ions then diffuse across the membrane, and this provides the energy for ADP to be phosphorylated and converted to ATP. So describe electron transport chain, which takes place in the inner mitochondrial membrane, and explain role in generating ATP. And in both these diagrams, which show the same thing that's happening, we can see that, first of all, NADH, which is reduced NAD, and FADH2, which is reduced FAD, first get oxidized, and this releases electrons to proteins in the electron transport chain. So those are the proteins, the electron carrier proteins on the membrane. And oxidizing these molecules also releases hydrogen ions. The electrons release energy as they pass down the electron carrier proteins in the electron transport chain. And this provides energy to pump hydrogen ions from the matrix to the intermembrane space. The hydrogen ions diffuse through ATP synthase channel, which is an enzyme on the membrane, by chemiosmosis down the hydrogen ion concentration gradient. This provides the energy for ATP to be made as hydrogen ions flow to the matrix, and this is known as oxidative phosphorylation, where ADP is phosphorylated with an inorganic phosphate and converted into ATP. The electron that is transported from one electron carrier protein on the membrane to another, as it moves along, it then gets finally accepted by an oxygen. So therefore oxygen acts as the final electron and proton acceptor, and this will then form water with the available hydrogen ions. Explain the importance of mitochondrial membrane. Now we can see that there is an outer membrane and an inner membrane on the mitochondria, and the proteins on the outer membrane allow pyruvate to pass through to matrix for the link reaction, whereas the inner membrane contains embedded proteins, and we know that these proteins are involved in the electron transport, where the electrons are passed from one electron carrier protein to another. And the other proteins that can also be found on the membrane of the inner membrane of the mitochondria is the ATP synthase channel, where ADP is phosphorylated to ATP, and the roles of all of those proteins in the inner membrane is oxidative phosphorylation, chemiosmosis to synthesize ATP. And here we can see the animation again, where we've got the electron transport chain. We start off with reduced NAD and reduced FAD, which then get oxidized, releasing hydrogen ions and electrons. The hydrogen ions move across the membrane. Electrons are then passed along from one electron carrier protein to another, and it provides the energy to pump hydrogen ions across the membrane from the matrix to the in between the two membranes. The electron is finally accepted by oxygen, so oxygen is the final electron acceptor, with hydrogen to form water, and the hydrogen ions then diffuse down a concentration gradient in ATP synthase, which provides the energy to phosphorylate ADP into ATP. Note the different names of these different parts. We've got the electron transport chain, where electrons are transported from one electron carrier protein to another. We've also got chemiosmosis, and this is where the hydrogen ions diffuse down the concentration gradient through ATP synthase. And then there is oxidative phosphorylation, where the electron carrier molecules NAD and FAD, which have been reduced, are then oxidized, and this finishes off with the phosphorylation of ADP by ATP synthase, which is why it's called oxidative phosphorylation. We should state that cellular respiration yields ATP, and that ATP can then be used as a source of energy for metabolic reactions, and those metabolic reactions also generate heat energy. Explain difference in ATP yield from one molecule of hexose sugar, such as glucose, in aerobic conditions compared to anaerobic. So if we're comparing aerobic and anaerobic respiration, we can see that the location of aerobic respiration starts off with cytoplasm, where glycolysis occurs, but then you've got the link reaction and Krebs cycle in the matrix of the mitochondria, and oxidative phosphorylation also occurring in the inner membrane. Whereas anaerobic respiration only occurs in the cytoplasm. For aerobic respiration, you've got the complete oxidation, complete breakdown of glucose, whereas it is incomplete or partial for anaerobic respiration. The final electron and hydrogen acceptor is oxygen in aerobic respiration, but due to the amount of reduced NAD that is made, pyruvate is then used as the final electron acceptor, because it then oxidizes those reduced NAD molecules. The products of aerobic respiration are carbon dioxide and water, where it's lactate in anaerobic respiration, which can dissociate to hydrogen ions and lactic acid. Both aerobic respiration and anaerobic respiration uses two ATP molecules at the beginning in glycolysis, but the amount of ATP made in aerobic respiration is somewhere between 30 to 38, depending on the condition, per molecule of glucose, whereas it's only 4 in anaerobic respiration. So you can see there's a big difference in the amount of ATP that is made comparing aerobic and anaerobic respiration. So in anaerobic respiration, the electron carriers NAD and or FAD are reduced in glycolysis and Krebs cycle, and the whole purpose of reducing them is to then produce many ATP molecules in oxidative phosphorylation. However, in anaerobic respiration, due to the lack of oxygen as a final electron acceptor, this results in pyruvate oxidizing reduced NAD or NADH. So no further ATP is produced other than the net total of 2 ATP molecules, because we used 2 ATP molecules and 4 was made, so the net amount of ATP molecules made is 2, and this is in glycolysis per 1 glucose molecule. So therefore in anaerobic respiration you've got partially broken down glucose, which is lactate, which is still rich in energy because it hasn't been fully oxidized. When there is finally enough oxygen available, that lactate can then be oxidized into pyruvate, and pyruvate can then enter the link reaction and Krebs cycle stage of aerobic respiration. (5.2) Photosynthesis. Photosynthesis occurs in both plants and algae, and it's an endothermic reaction, which means that it requires energy for the reaction to occur, and that energy comes through light energy that is absorbed to react carbon dioxide and water, which produces glucose and oxygen, which is why plants and algae are producers. And here you've got the word equation: carbon dioxide reacts with water, which are the reactants, to produce glucose, and oxygen is also a byproduct. And we see it's called photosynthesis, is you're synthesizing glucose using light energy. There are two types of spectrums related to photosynthesis. We've got the absorption spectrum and the action spectrum. In the absorption spectrum, it shows the absorption of light for different wavelengths, and this only involves wavelengths. So you can see there are lots of different types of photosynthetic pigments that plants and algae can have, and we can see what are the different absorption of light for those different wavelengths. So for blue light or green light or red light, we could then compare the amount of light that is absorbed on those different wavelengths. Whereas in action spectrum, that shows the rate of photosynthesis at each wavelength. So this only involves photosynthesis or oxygen production. So therefore, what's the total rate of photosynthesis at blue wavelength or green wavelength or red wavelength? This has not got to do with the different types of photosynthetic pigment. It's more the overall rate of photosynthesis at each of the wavelength, which clearly would be a combination of the different photosynthetic pigments. Explain why many plants have a variety of different photosynthetic pigments. Why would there be different types? The most common one you would have heard of is chlorophyll, but there's actually two types of chlorophyll, A and B. Having different pigments means that you could absorb light at different wavelengths for photosynthesis, because the different photosynthetic pigments absorb different wavelengths or range of wavelengths, and therefore having different combinations of them means that you could absorb more light at different wavelengths. So chlorophyll A and B absorbs at the lower and higher ends of the visible spectrum, which is more similar to the blue and the red parts of the wavelength. Accessory pigments such as carotenoids, they absorb other wavelengths, and this enables plants to absorb more of the visible spectrum. State how action and absorption spectrum show chlorophyll is used in photosynthesis. So we can see if we were to pour all the photosynthetic pigments absorption of light together, we'd get a similar shape to the action spectrum. So the spectra are similar, they overlap. The peaks and troughs follow a similar pattern. So an example of this would be there's a similar peak at blue or red, and obviously there's a similar trough at green. Core Practical 10. Investigate effects of different wavelengths of light on rate of photosynthesis. Oxygen is a waste product of photosynthesis, so one way to investigate the different wavelengths of light on the rate of photosynthesis is to compare the amount or rate of oxygen produced per minute. So you can measure the rate of oxygen production, and that could be used as an indicator of the rate of photosynthesis. And this could be done using an equipment similar to shown in the diagram here, where you've got a syringe which shows the volume of oxygen produced, and that could be compared to time, the amount of time it takes to find out a rate of photosynthesis. So the more photosynthesis a plant or algae does, there should be a larger volume of oxygen per time. So the independent variable could be the wavelength of light. We could use different wavelengths, and they can be changed by using colored filters. The dependent variable, as explained, was the volume of oxygen collected, and that would be done using a syringe, which is a lot more accurate. The control variables, therefore, would be the mass of the plant. Temperature will also be a control variable because that affects the rate of photosynthesis. That will be the same as the carbon dioxide concentration, and that could be done by using sodium hydrogen carbonate in the water. Describe what was measured in an investigation where the rate of photosynthesis was expressed as micromoles per meter squared per second. So for the micromoles, that could be the carbon dioxide used or oxygen stroke GP stroke gout stroke glucose made. Per meter squared could be the area of the leaf or the area of open stomata, and then per second is the amount of time that was used. So if it was done over 120 seconds, the results would be divided by 120 to make it per second. Core Practical 11. Investigate presence of different chloroplast pigments using chromatography. So as I mentioned earlier, chlorophyll is not the only photosynthetic pigment, and there are actually two different types of chlorophyll, and each photosynthetic pigment absorbs different ranges of wavelength. And we could then separate these photosynthetic pigment using chromatography. So first you add pigment extract to the start line. You then concentrate the spot by dry and repeat to evaporate the propanol solvent. Therefore you have a small concentrated spot of extract that builds up, and that prevents the extracts from spreading out when you put it into the solvent. You place the paper in a propanol solvent with the start line and spot above solvent. So therefore, as the solvent moves up, it then reaches the start line and the spot. You then obtain a solvent front when the solvent reaches near the top, and that could be used to calculate the RF value, which is the distance traveled by the pigment divided by the distance traveled by the solvent. The RF values can then be compared to known pigments in the same solvent, plus it could be used to check the pigment color to identify the different types of photosynthetic pigment. So therefore you could identify what the different photosynthetic pigments are in different plants by comparing the RF value and also looking at the color of the pigment of the chromatogram. Explain how cyclic and non-cyclic photophosphorylation produces ATP, reduced NADP, and oxygen. So here we've got a part of the chloroplast, and to start off with, in the thylakoid membrane, chlorophyll of photosystem 1 absorbs light energy. That light excites chlorophyll electrons to high energy levels, which releases the electrons. The electrons then pass down electron carrier proteins in the electron transport chain, and that releases energy, and that energy can then be used to pump hydrogen ions from the stroma to accumulate the hydrogen ions in the thylakoid space. The hydrogen ions in the thylakoid space can then move through the ATP synthase channel, which is known as chemiosmosis, and this results in the photophosphorylation, which is the phosphorylation of ADP to ATP. So ATP is therefore produced. The electron that is passed down from one electron carrier protein to another in the electron transport chain can then do two things. It could either return back to photosystem 1, which is known as cyclic phosphorylation, and that electron can therefore be excited again in the future, or the electron can then be given to NADP. So NADP becomes reduced, and this is a non-cyclic phosphorylation. So that electron therefore is not available again in the future to be excited. The reduced NADP and the ATP that is produced, both are then used in the Calvin cycle. The electrons from photosystem 2 can be replaced if they are provided to reduce NADP, and they can be replaced those lost by photosystem 1. And this occurs because there is photolysis of water. In other words, water is split using light, and when it's split, it provides electrons to replace those lost by photosystem 2. Photolysis of water also results in the production of oxygen, which is a byproduct of photosynthesis. Describe structure of chloroplast. So here we can see the structure of chloroplast on the top right, and we can see that it has both an outer and inner membrane, and this is known as the chloroplast envelope. The space within the chloroplast is known as the stroma, and here light-independent stage of photosynthesis occurs. Chloroplast also contains grana, which is a stack of thylakoids, and those stack of thylakoids are connected to each other by a lamella. They connect the grana, and this increases the surface area so that there is more light absorption. Describe role of thylakoid membranes in photosynthesis, where those thylakoid membranes compartmentalize the stroma. It's the site of where light-dependent reactions occur because it contains chlorophyll, also contains electron carrier proteins for the electron transport chain. And the membrane increases the number of pigments or proteins that can be held for ATP to be synthesized. It also contains ATP synthase. Explain light-independent stage of photosynthesis. So in the light-dependent stage, we reduce some NADP and we also produce some ATP, and both of those are then used in the light-independent stage of photosynthesis. To start off with, carbon dioxide diffuses into the stroma, and that carbon dioxide binds to a 5-carbon ribulose biphosphate, or RUBP, using the enzyme Rubisco. And this means that carbon is fixated. This results in the formation of two 3-carbon glycerol 3-phosphate molecules. The glycerol 3-phosphate molecule is reduced to synthesize 3-carbon GALP, which is glyceraldehyde phosphate, using reduced NADP and the energy from ATP, and both of those were produced in the light-dependent stage. So the reduced NADP and the energy from ATP causes glycerol 3-phosphate to be reduced, and that means that you've now produced GALP. That gout molecule could be used to regenerate the 5-carbon ribulose biphosphate in the Calvin cycle so that this cycle continues. However, some of it is used to synthesize 6-carbon glucose. So here you could see an animation of the Calvin cycle where you start off with carbon dioxide diffusing to the stroma. It then binds with IUPP for the carbon to be fixated using an enzyme Rubisco. This causes the formation of glycerol 3-phosphate, and then that glycerol 3-phosphate becomes reduced using reduced NADP and energy from ATP, both provided by the light dependent stage, to form gout, and the gout can therefore be used in the cycle to form IUBP so that the Calvin cycle continues, but some of that gout could then be used to produce glucose. Explain how gout is used as a raw material. So we know one of the things it's used for is to produce glucose. However, you could produce other materials that will increase the biomass of plants and algae. So to start off with, gout could be used to produce cellulose, and that is used to strengthen the cell wall. So cellulose is a polysaccharide that we learn in topic 1. It could also be used in respiration because it could be used to make glucose, and that glucose is then used to make ATP. Gout could also be used to make oils and fats, which are collectively known as lipids, and that's the use of storing energy. Another raw material of gout is proteins, so you can synthesize those proteins because gout could be converted to amino acids, which then can be joined together to produce polypeptides, which fold into proteins based on their structures. Gout could also be used to produce starch, which is a storage of energy, and it could also be used to convert into sucrose, which is a way of transporting sugars. Another molecule that gout could be used to produce are nucleotides for both DNA and RNA. Which factors affects the rate of photosynthesis? So in GCSE we learned about 3 different factors. We've got light, carbon dioxide, and temperature. So we know what a limiting factor is, and you can see in this diagram we've got someone who's limiting factor in terms of their growth. So a limiting factor is a factor that limits the rate of photosynthesis. So on this diagram we can see light is a limiting factor at the beginning of the reaction because there is a low intensity of light, which means that the rate of photosynthesis is low. So the reason why the rate of photosynthesis is low is because there's not enough light intensity. By increasing the light intensity, we can see that the rate of photosynthesis increases. So what is limiting the rate of photosynthesis at the beginning of this reaction? It must be the light intensity or the low light intensity. Explain limiting factors that limit rate of photosynthesis. So as we mentioned, when light intensity increases, the rate of photosynthesis also increases. Higher light intensity increases the amount of chlorophyll that is excited in the light dependent stage of photosynthesis, and therefore the amount of NADPH or reduced NAD and ATP produced in the light dependent reaction also increases. However, at low light intensity, light is the limiting factor because there is insufficient reduced NAD and ATP produced, for reactions of the light-independent stage. So those two molecules that are made in the light-dependent stage, not a lot of them would be made, and therefore there's a limited amount of that for the light-independent stage, where glycerol 3-phosphate is converted into GALP. And that is why the rate of photosynthesis is low when the light intensity is low. When the light intensity is very high, the rate of photosynthesis doesn't increase anymore, and this is because something else is a limiting factor. So clearly that could either be carbon dioxide concentration or temperature. When carbon dioxide concentration increases, the rate of photosynthesis also increases, and that's because there's more carbon dioxide reactant available for carbon fixation in the Calvin cycle. However, at low levels of carbon dioxide, the carbon dioxide is a limiting factor. And just like light intensity, when carbon dioxide concentration is very high, the rate of photosynthesis doesn't increase. There's something else a limiting factor. So in this case, light and or temperature might be the limiting factor when there is a high concentration of carbon dioxide. For temperature, this is linked to enzyme. So when temperature increases, the rate of photosynthesis increases, and that's because higher temperature increases kinetic energy for successful collision between enzymes and substrate involved in the light-dependent and Calvin cycle reactions. However, at low temperatures, temperature is a limiting factor. So you can see that there. By increasing the temperature, the rate of photosynthesis will increase. When temperature is too high, above the optimum, the rate of photosynthesis decreases because enzymes involved in the light-dependent and Calvin cycle will denature, and therefore the rate of photosynthesis will decrease. Here we can see a summary of both aerobic respiration and also photosynthesis. So on the top, we start off with glycolysis, which happens in cytoplasm. If there is presence of oxygen, we know that the product of glycolysis, which is pyruvate, then moves through the membrane into the matrix of the mitochondria, where both the link reaction and the Krebs cycle occur, and then the reduced NAD and reduced FAD produced are all used in oxidative phosphorylation, which happens in the inner mitochondrial membrane, and that produces ATP. Whereas in photosynthesis, we've got the light-dependent reaction, which happens in the thylakoid membrane, and the light-independent reaction that occurs in the stroma. And the whole point of the light-dependent reaction, if it is non-cyclic phosphorylation, is to reduce NADP and ATP, which can then be used in the Calvin cycle of the light-independent reaction to convert glycerol 3-phosphate into GALP, and that GALP can therefore be used as a raw material to make glucose and other materials as well. Compare and contrast how ATP is synthesized in mitochondria and chloroplasts. In terms of their similarities, both use electron transport chain on the membranes, so therefore there are electron carrier proteins of the membrane where electron is passed from one carrier to another, and that loses energy or provides the energy for hydrogen ions to be pumped across the membrane, which then drives the production of ATP. They also have ATP synthase on the membrane, which is driven by the hydrogen ion concentration gradient or chemiosmosis, where the hydrogen ions diffuse down the concentration gradient, and that provides the energy for ADP to be phosphorylated into ATP. When making the ATP in mitochondria is oxidative phosphorylation, because you've got reduced NAD and reduced FAD, which are oxidized, and then you've got the phosphorylation of the ADP to ATP, whereas in the chloroplast is photophosphorylation, because the light energy is used to excite electrons in chlorophyll of photosystem 1 and 2, and those electrons then move from one electron carrier protein to another, which pumps the hydrogen ions across the membrane, and then with ATP synthase, that causes the ADP to be phosphorylated. So that all starts from light energy that excites electrons, so it's photophosphorylation. Where do the electrons start? Well, it's brought about by reduced NAD and reduced FAD in the mitochondria. However, in the chlorophyll, the light excites the electrons, and that's where they start in the chloroplast. In terms of where they end, oxygen is the final electron acceptor in the mitochondria. However, in terms of the chloroplast, it depends if it's cyclic or non-cyclic phosphorylation. So in non-cyclic phosphorylation, the electron eventually is used to reduce NADP, which is then goes to the Calvin cycle of the light independent stage, whereas if it's cyclic phosphorylation, where there is no NADP available, the electron then returns back to the chlorophyll of photosystem 1 or even photosystem 2. Explain why mutations that affect proteins involved in electron transport chain lead to buildup of lactate. So what are these proteins that are in the membrane that are involved in electron transport chain that can be mutated, and what effect would that have if the mutation occurs? Well, the mutation may prevent the electrons being passed down from one electron carrier protein to another, and because they're not being passed on, therefore you don't have hydrogen ions being pumped across. The other protein that could be affected is ATP synthase, where ATP is produced. And because of this, that means that NADH, or reduced NAD, can't be oxidized. So if it can't be oxidized because there's no oxygen available as the final electron acceptor, you therefore need the NADH, or reduced NAD, to be oxidized by something else, because they can't be used in the oxidative phosphorylation. So instead, pyruvate gets oxidized, and when pyruvate is oxidized, it produces lactate. That lactate is produced at a faster rate than it's removed. So in other words, it builds up. Explain why algae cells need to be illuminated at high light intensity during investigation on effect of carbon dioxide concentration on photosynthesis. So if we want to see how carbon dioxide concentration affects photosynthesis, we need to make sure that the light intensity is very high. Why do we need to make sure that happens? Because therefore the light intensity will not be a limiting factor, and the reason why you don't want light intensity to be a limiting factor is because light is involved in producing ATP and NADPH, or reduced NAD, which therefore is produced in the light dependent reactions. We don't want them to be a limiting factor, because we only want to see what effect carbon dioxide have. So carbon dioxide concentration should be the only limiting factor, so we could then see what the effect of carbon dioxide concentration is on photosynthesis, and not light intensity, or not ATP stroke reduced NAD. And that is the whole of topic 5, energy for biological processes, 5.1 cellular respiration, 5.2 photosynthesis.